Hello everyone. Today we will study one of the most important and classical example of gene regulation in prokaryotes, the tryptophan operon, commonly known as TRP operon. The operon is found in bacterium E. coli and serves as an excellent model for understanding how microorganisms regulate gene expression according to their metabolic needs. So before discussing the operon itself, it is important to understand why gene regulation is necessary. Living cells require energy for growth, reproduction, transport, and metabolism. If cells continuously produced all enzymes regardless of necessity, a large amount of energy and resources would be wasted. Therefore, cells have evolved mechanisms that allow them to synthesize proteins only when required. The tryptophan operon is one such regulatory system that controls the biosynthesis of amino acid tryptophan. Tryptophan is one of the 20 standard amino acids used in protein synthesis. Since it is essential for many cellular functions, bacterial cell must maintain an adequate supply of it. However, synthesizing tryptophan requires several enzymatic reactions and consumes significant cellular energy. Therefore, bacteria produce tryptophan only when it is not readily available in the environment. The tryptophan operon enables the cell to achieve this balance efficiently. A tryptophan operon is classified as a repressible operon. This means that under normal conditions, the operon remains active, allowing transcription of the genes required for protein tryptophan. It allows the transcription of the genes which is required for tryptophan synthesis. When tryptophan becomes abundant, the operon is repressed or switched off. Thus, the default state of the tryptophan operon is on, unlike the lac operon whose default state is off. So as we know that lac operon is an inducible operon. So the operon consists of several important components. First is the promoter, which is the DNA sequence where RNA polymerase binds to initiate transcription. Adjacent to the promoter is the operator, which serves as the binding site for the repressor protein. Following the operator is the leader region, also known as a TRPL, which plays a critical role in attenuation. Beyond the leader sequence lie the 5 structural genes TRPE, TRPD, TRPC, TRPB, and TRPA. These genes encode enzymes required for converting choramic acid into tryptophan through a series of biochemical reactions. Separate from the operon is the regulatory gene TRPR. This gene produces a protein called the TRP repressor. Interestingly, the repressor protein synthesized by TRPR is initially inactive and cannot bind to the operator. For the repressor to become functional, it requires interaction with tryptophan molecule. Let us now examine what happens when the concentration of tryptophan inside the bacterial cell is low. Under these conditions, the cell requires more tryptophan for protein synthesis and other metabolic processes. Since tryptophan molecules are scarce, they cannot bind to the repressor protein. Consequently, the repressor remains inactive and is unable to attach to the operator region. Because the operator is free, RNA polymerase binds to the promoter and begins the transcription of the structural gene. Polycistronic mRNA containing the coding sequences of TRPE, TRPD, TRPC, TRPB, and TRPA is produced. This mRNA is translated into enzymes that catalyze the biosynthesis of tryptophan. As a result, the intracellular levels gradually increase. Thus, when tryptophan is deficient, the operon remains active and enzyme production continues. Now let us consider the opposite situation where tryptophan is present in excess. When the cell accumulates sufficient quantities of tryptophan, the amino acids itself act as a co-repressor. A co-repressor is a small molecule that binds to an inactive repressor and converts it into active form. In tryptophan operon, the tryptophan molecules bind to the tryptophan repressor protein and induce a conformational change. This structural alteration enables the repressor to recognize and bind specifically to the operator sequence. Once the activated repressor co-repressor complex binds to the operator, it physically blocks RNA polymerase from transcribing the structural genes. Since transcription cannot proceed, no mRNA is produced and no enzymes for tryptophan biosynthesis are synthesized. Consequently, the cell stops producing tryptophan. This mechanism ensures that energy is not wasted synthesizing an amino acid that is already available in adequate amounts. This type of regulation is known as negative feedback inhibition because the end product of pathway inhibits its own synthesis. Although repression provides an effective means of control, bacterial cells require an even more sensitive mechanism to fine-tune gene expression. This additional level of control is called attenuation. Attenuation is a unique regulatory mechanism found in several amino acid biosynthetic operons and is specially well characterized in tryptophan operon. So to understand attenuation, we must remember that in bacteria, transcription and translation occur simultaneously. As RNA polymerase synthesizes mRNA, ribosomes can immediately begin translating the newly formed transcript. The leader region of tryptophan operon contains a short peptide coding sequence with two consecutive tryptophan codons. It also contains four complementary RNA regions capable of forming alternative secondary structures. The behavior of the ribosome during translation of the leader peptide determines which RNA structure forms when tryptophan levels are high. Charged tRNA molecule carrying a tryptophan are abundant. The ribosome rapidly translates the leader peptide without pausing. This allows the region 3 and 4 of the mRNA to pair with each other, forming a stable terminator hairpin loop followed by a sequence of uracil residues. This structure functions as a transcription terminator. As a result, RNA polymerase detaches from the DNA template before reaching the structural genes. Transcription is terminated prematurely and the biosynthetic enzymes are not produced. In contrast, when tryptophan levels are low, charged tryptophanyl tRNA molecules are scarce. As the ribosome encounters the two tryptophan codons in the leader peptide, it stalls because insufficient tryptophan-loaded tRNA is available. This stalling changes the folding pattern of the mRNA. Instead of regions 3 and 4 pairing, regions 2 and 3 pair together to form an anti-terminator structure. Because the terminator hairpin cannot form, RNA polymerase continues transcription through the structural genes. The enzymes required for tryptophan synthesis are subsequently produced, allowing the cell to replenish its tryptophan supply. Therefore, tryptophan operon possesses two highly coordinated regulatory system or mechanism. The first mechanism, repression, determines whether transcription can begin. The second mechanism, attenuation, determines whether transcription should continue after initiation. Together, these mechanisms provide precise control over gene expression and ensure that the cell responds rapidly to changing intracellular concentrations of tryptophan. The biological significance of the tryptophan operon is immense. It conserves cellular energy by preventing unnecessary enzyme synthesis, maintains amino acid homeostasis, and enables bacteria to adapt efficiently to nutritional changes. Furthermore, studies of the tryptophan operon have contributed significantly to our understanding of molecular genetics, transcriptional regulation, and the relationship between the genotype and phenotype. So in summary, the tryptophan operon in E. coli is a repressible operon responsible for controlling the biosynthesis of tryptophan. It consists of a promoter, operator, leader sequence, and 5 structural genes. The regulatory gene tryptophan R produces an active repressor that becomes active only when bound by tryptophan, which acts as a co-repressor. High level of tryptophan represses transcription, whereas low level permit transcription and enzyme production. In addition, attenuation provides a second layer of regulation through alternative RNA secondary structures formed in the leader region. Together, repression and attenuation creates a highly efficient regulatory system that serves as one of the most elegant examples of gene control in bacteria and remains a cornerstone of molecular biology education and research. Thank you.